guys, let's do just a little bit of review from yesterday before we start in on the new lesson. So the first question, how is the finished polypeptide removed from the ribosome? So remember, when we get to that stop codon on the mRNA, the stop codon, instead of creating an amino acid, is going to code for this release factor. And so that release factor is going to cause the cut or the hydrolysis of the polypeptide strain to free the polypeptide strain. It'll exit through the exit tunnel, which is what you see here, and then this translation initiation complex will disassemble, and the peptide will be released to go on to to its destination, essentially. Okay, so it's removed by the release factor that is synthesized with the stop codon, essentially. The second question, what determines where a specific protein is made? So remember, yesterday we talked about unbound ribosomes and bound ribosomes. Those unbound ribosomes are in the cytoplasm. The bound ribosomes are bound on the rough ER. Okay, so these are going to have some types of modifications, and they're generally going to be sent outside the cell. These other ones are going to be a little simpler, and they are going to stay inside the cell. Okay, and so in this process, we have this signaling peptide that's going to signal if that protein needs to be made in the rough ER. And if it is, then the signal recognition particle will recognize it, target it to the ER, and it will continue its synthesis there. So that is determined by where the protein is being made and any type of modifications it has. And then the last one, draw the general structure of the amino acid. I've drawn this in a couple of lessons, both virtual and in person. So hopefully we are getting close to being able to do this. That one should be a single bond. Sorry, I screwed that up. And like this. And there's our R group. Here's our carboxyl group. And here is our amino group. Okay, and if you drew the OH at the bottom and the double bond to the O on top, if you flip-flopped those two parts, that would be perfectly fine. Okay, but that should be a single bond. That shouldn't be a double bond right there. All right, so that is the general structure of an amino acid. So we talked about that in biochemistry, but I don't think many of you all remembered it from then. Okay, so today's lesson is a little bit shorter. Today we are talking about mutations, but let's just take a step back before we do that and go through this whole transcription and translation process that we have been talking about in this unit for the last week and a half or so, maybe longer than that. So in transcription, remember, transcription is happening in the nucleus. Okay, we're right here in the nucleus. We are talking about eukaryotic cells for right now. Okay, the DNA is going to be converted into an mRNA transcript. So we're reading the DNA on the 3' to 5', and we're making the RNA in the 5' to 3' direction. Okay. We've got this major enzyme that's going to do all of the work. That's that RNA polymerase. Okay, so once we get all of that done, that DNA copied into mRNA, we've got that primary transcript. It has to undergo RNA processing. And so what happens in our RNA processing, we're going to add a 5' cap. We're going to add this poly A tail that you see here. Those are important because they are going to help with the binding on the ribosome in translation. They're also going to protect the mRNA from degradation once it goes into the cytoplasm. Okay? The introns are going to be spliced out and the exons are going to be pushed together. That's the job of the spliceosome. That's all still happening in the nucleus for RNA processing, okay? Now, once we get into the cytoplasm for the next step, we have translation. So in translation, that's going to happen on the ribosome. We're going to take this mRNA and we are going to translate it into this polypeptide that you see here, okay? So we've got our tRNAs that are helpful there. The tRNAs are going to bring in the amino acid as coded for on the mRNA, okay? In our RNA, we have these three, the ribosomal RNA. We have, to bring all of those pieces together to get it started, the 60S subunit and the 40S subunit, or the large and the small subunits, to make that translation initiation complex. Then we bring in these tRNAs. The anticodon is complementary to the codon on the mRNA, and it has the amino acid that is coded for by the mRNA, okay? Once that comes in here into the A spot, this guy is going to be hooked on top of here. The polypeptide is going to hook on top of the A spot. The A will shift over to the P spot. This empty guy, once this is cut off, will be empty. He'll go over here, and then the previous E will be kicked out. And then we'll bring in the next amino acid read on the next sequence of the mRNA, okay? Now, these amino acids are created by this enzyme that we talked about yesterday, aminoacyl tRNA synthetase. It attaches the amino acid to the tRNA and makes it charged, okay? Once that amino acid is attached, it's called charged, okay? And so once we finish with this translation piece, we will have that growing, that polypeptide chain. We'll stop once we get to the stop codon, somewhere down here, and then it'll code for those release factors that'll cut this polypeptide free and send it on its way, okay, assuming it doesn't need any other modifications. All right, so that's kind of the big picture that we've been discussing. So let's talk about mutations today. So you probably already know what a mutation is. A mutation is any change in the genetic information in the cell. So we're talking about genetic information. That could be DNA or RNA. If you have the DNA that has a mutation in it, that's a mutation. But you also can have the RNA that gets mutated. So mutations can happen in DNA replication. They can happen in transcription. Less likely to happen in translation, more likely to happen in replication or transcription. But mutations are actually important because they are the ultimate source of new genes. Mutations give us genetic variation. And in a few weeks, we will get into our evolution unit, and you will see that genetic variation is what drives evolution. It's how we get new species. And so mutations are an important part of that. So what I want you to realize is there's really two basic type of mutations. There are chromosome mutations, and we talked about those in the last unit, and there are gene mutations. Chromosome mutations are things like Down syndrome. They're things that you can see in a karyotype. They're big pieces of a chromosome that have been mutated. You get one less chromosome or one more chromosome. That's aneuploidy, remember? Polyploidy is a whole extra set of chromosomes, which that doesn't really happen in humans. You can have a deletion of a section of a chromosome, or a translocation of a chromosome where parts of them kind of switch. But those are big mutations. Those are going to have major effects, okay? Now, today what we're talking about is gene mutations. So these are a smaller change in a nucleotide. You cannot see these on a chromosome. Now, they can have negative effects, and they can absolutely cause, bad things too, but generally they're not quite as severe as a chromosomal mutation, okay? So let's talk about these and go through some of the types. So hemoglobin, if you may know hemoglobin is a protein that's in your blood. And so normal wild-type hemoglobin, it's a series of several amino acids, but one part of it reads CTT on we're going to call this one, we're going to reference this top strand, okay? But in a sickle cell hemoglobin, there's been a mutation, and CTT is switched out into CAT. So that change in that one amino acid causes a sickle cell hemoglobin and sickle cell anemia. So normal red blood cells are nice and round, okay? Sickled blood cells look like a banana, okay? Remember, red blood cells are, as they remember, we haven't really talked about this, not really part of our content, but red blood cells are important for carrying oxygen. And so, as you can imagine, sickled red blood cells don't carry oxygen very well. They don't have that nice surface area to volume ratio like we talked about in cells. Remember, you increase the surface area and with the same volume, you increase the surface area to volume ratio. It makes it more efficient at doing its job. And so sickled cells don't have that. They also don't move through the blood vessels very well. They tend to kind of cause traffic jams in the blood vessels, which can lead to something that's known as sickle cell crises. They're just very painful episodes that sickle cell patients have. And so, in the normal hemoglobin molecule, this is going to code for glutamine, and in the sickle cell hemoglobin, it's going to code for valine. So one amino acid was changed, and it's going to result in that genetic disorder, okay? And then that can be passed down to the offspring, okay? So this is an example of a point mutation. It was changed in one place, okay? So there are a few types of point mutations that you can have: a substitution, an insertion, and a deletion. So this is an example of a substitution. You see the T was substituted for the A, okay? So substitution, replacement of one nucleotide with another nucleotide, okay? I'm going to come back to this in just a second. Let's look at... you can also, let's go ahead and go back. A silent mutation. A silent mutation is when you have one codon that causes the same amino acid to form. I mentioned this in a lesson last week. So let's say, I'm making this up so I don't know. Let's say GCC makes the amino acid serine, but also GCA makes the amino acid serine. Okay, you were supposed to make GCC, we'll say that was normal, but you had a mutation and your body made GCA. And so this is a silent mutation. You did have that change, but it resulted in the same thing. The same amino acid was formed. And so it has no effect on the phenotype. That's a silent mutation. Okay? Now, a missense mutation changes one amino acid to another. Okay? That's what we see here. This is a point mutation with the hemoglobin, but it's also a missense mutation because it changed this glutamine to a valine. That's a missense mutation. Change one amino acid to another, may have little effect on the protein. So it does affect the protein. It makes the wrong hemoglobin. It does cause problems, but it's not the end of the world, so to speak. And I don't mean that lightly, but it is a missense mutation. There's still that protein. It's still going to work. It's just not going to be quite as effective, okay? Now, next is a nonsense mutation. So a nonsense mutation is when you have an early stop codon. Let's talk about another type of point mutation. Remember, we said point mutations could be a substitution, an insertion, or a deletion. So insertions and deletions also are categorized as a frame shift mutation. And that's because it shifts the reading frame, okay? So in an insertion, we add an extra amino acid. In a deletion, we take away an amino acid. So the last thing here, mutagens and carcinogens. So there are several agents that interact with DNA and cause mutations, and those things are called mutagens. So for an example, UV light is an example. We talked about in a previous lesson quite a while ago that UV light can sometimes cause thymine dimers, cause the thymines that are side by side to pair together and buckle, and changes the structure of the DNA molecule. Sometimes UV light can also cause DNA to degrade, and so that's an example of a mutagen. Carcinogens are things like smoking, and they can cause cancer. So often these two are linked. Most mutagens are carcinogens, like we know UV light to cause cancer, but also most carcinogens, such as smoking, are mutagens because they interact with the DNA and can result and cause mutations. So any type of genetic disorder that's any type of mutation that you are born with is considered to be a genetic disorder. Now, people can be born with mutations that they are not predisposed to genetically. So maybe your mom is normal and your dad is normal, but the child is born with cystic fibrosis. They're not carriers for it, anything like that. So what that means is that was just a random mutation, and that can still happen, okay? You can still have mutations that happen like that. Sometimes mutations are good, but generally they're going to result in a faulty protein, okay? So today I want you to take a look at this assignment. It should be in your binder. How bad is the mutation? So normally when we read this sentence, we would read, hey, did Max the fat cat eat the rat? And you could write those in these boxes here. Hey, did Max, and so on and so forth, okay? So that's what you're going to do. Now, this first example, it's a silent mutation. So we say, hey, did Max, okay, and so on, the fat cat eat the rat. It's silent because it changes it. There is a mutation, but it still basically makes the same thing. The sentence reads the same, okay? Just like if we made the same amino acid, but we changed the bases that did so, okay? Now, a missense mutation, hey, did Max the fat cat eat the bat, okay? So that's a missense mutation. So it changed it. The protein is not the same, the sentence is not the same, but it's pretty close, right? It's pretty close. That protein's gonna work, maybe not quite as well, but it'll do something, okay? In the next one, this is gonna be, hey, and then there's an insertion. So this is a frame shift mutation. So now, it's gonna really screw that up. Hopefully through this example, you see what I mean by a frame shift. It shifts the reading frame. You don't read the same groups of 3 letters to be anymore, okay? Nobody can make anything out of that. That amino acid or that protein is gonna be so screwed up, that's not gonna work very well, okay? A deletion, okay, is also gonna be a frame shift mutation because you're gonna have something like this, okay? And so, yeah, I think that's right. I don't know. I maybe missed some of those letters, but you can figure it out, okay? And so that's a deletion. Now, you could also have a deletion downstream. And so we're going to take out THRAT. And so it messed up a few of the amino acids, But the beginning part, hey, did Max the fat cat eat, is still normal. It's just the rat that got screwed up. And so that deletion would have less of an effect than this deletion before it. Okay? This is going to have a greater effect. That's going to have a greater effect. This is going to have a lesser effect just because of the location of the mutation. Okay? Now, you can also insert a codon, so a whole group of letters. Hey, you, did Max the fat cat eat the rat? And so that's going to change it a little bit. And then you can also delete a codon. So that's just a few examples of how bad is the mutation. So complete this and think about those different types of mutations and upload that to Google Classroom. And if you have any questions, then let me know. Have a great day.